It is one of those topics that if you spend enough time around medical textbooks or, surgical wards, just sort of accept it as a brute fact of life.
Hmm.
Colorectal cancer is common. Here are the stages. Here is the survival rate. But if you actually stop and look at the biological machinery underneath it all,
Oh, it's a mess.
it is a mess. It is a chaotic, complex mess.
It is a beast. And honestly, looking at the stack of notes we have for this deep dive, it seems like this particular topic, colorectal adenocarcinoma, is where a lot of people just hit a wall.
They really do.
We have gene acronyms that all sound the same.
Yep.
We have signaling pathways that seem to run backwards sometimes. We have different types of instability. It's just a lot.
It feels like a cognitive bottleneck. I like that phrasing you used in the pre-show.
Yeah.
It feels accurate because for so many learners, whether they're medical students, surgical residents, or frankly just insanely curious minds, this is where the understanding usually collapses.
Exactly. Most people can memorize the staging. I mean, the TNM system tumor, node, metastasis, that's logical enough.
It's basically geometry and geography.
Right. How big is it and where has it gone?
Yeah.
They can even memorize what a tumor looks like on a glass slide.
Mm.
But the pathway logic,
Mhm.
the actual narrative story of how a healthy cell decides to go rogue and become a tumor, that's where people get completely stuck.
Which is exactly what we're going to fix today.
I hope so.
Our mission for this deep dive is to move away from rote memorization. We're going to embrace what our source material calls mechanistic storytelling. We want to understand the plot of the movie, not just the cast list.
That is the goal. Because in colorectal cancer, it really isn't just one plot. It's not a single story of cell breaks, cancer grows.
Yeah.
There are specifically 3 distinct storylines, 3 different biological roads that all, unfortunately, lead to that same crash. And if you just try to memorize the names of the genes involved, APC, KRAS, MSH2, BRAF, it just becomes this alphabet soup of confusion.
You'll mix them up. You'll absolutely forget which one is the brake and which one is the gas pedal.
Precisely. So we need to structure this properly.
So here's the plan. We are going to break this down into layers. First, we have to define the molecules, our cast of characters, so to speak.
Right.
We need to understand what they do when they aren't causing cancer. What's their day job? And then we're going to watch them break one by one.
And the best part is, once you understand the mechanism, you don't really have to memorize the textbook definitions anymore.
No, you can just see it.
You can just visualize it happening, and we've got some really solid memory hooks to help with that.
But before we get into the nitty-gritty of the genes, the source material highlights one core question that frames absolutely everything. It suggests that if you ever feel lost in the weeds of molecular biology, you just come back to this one dichotomy.
It really is the anchor for the whole subject.
Yeah.
I mean it. If you're looking at colorectal cancer, you have to realize it develops in 2 main ways.
Okay.
You either lose the brake system slowly, or you lose the spell checker suddenly.
Losing the brakes slowly or losing the spell checker suddenly. That's a great way to put it.
That is the fundamental divide. Everything else we talk about today just hangs on that. The broken brake model, that's the APC pathway. That's the classic route, the one you read about most.
Right.
The spell check failure, that's the AMPHOGS pathway. And then there's the third one, the CMMP pathway, which is a bit more like a hacker getting into the car's software, but we will definitely get to that.
Okay, I love that setup. It gives us a map. We have a car crash caused by bad brakes, and we have a disaster caused by bad instructions.
Exactly.
But before we can understand the crash, we need to know how the car actually works. We need to meet the players.
Let's introduce the cast.
All right, segment 1, the molecular players. We're going to walk through the key proteins and genes. I want to know what it is, what it normally does, and I want that memory hook you mentioned. Let's start with the big one, APC.
APC. This stands for Adenomatous Polyposis coli. It lives on chromosome 5q21, if you want the address.
Yeah.
This is the heavyweight champion of colon cancer biology. I mean, if you understand APC, you understand something like 80% of these cancers.
Okay, so 5q21 is the address. But what's its job? What does it do on a Tuesday in a healthy colon cell? Is it a builder? A destroyer?
It is a tumor suppressor, but specifically, its job is to form what's called a destruction complex.
A destruction complex. That sounds incredibly aggressive for a healthy cell.
It is, but it's necessary aggression.
Right.
APC is basically a garbage disposal unit.
A garbage disposal.
Its entire purpose in a normal, healthy cell is to hunt down another protein called beta-catenin.
beta-catenin.
Yep. APC works with some other proteins to grab beta-catenin, tag it for destruction, a process we call ubiquitination, and then it sends it to the cellular shredder.
The shredder being the proteasome.
Exactly. So in a healthy cell, APC is constantly working. It's vigilant. It keeps the levels of free-floating beta-catenin extremely low in the cytoplasm. It's basically population control for this one protein.
So if APC is the garbage disposal, what happens when it breaks? What if the disposal unit gets jammed?
The garbage piles up.
Simple as that.
Simple as that. Specifically, beta-catenin piles up. And when beta-catenin accumulates, it doesn't just sit there. It travels into the nucleus of the cell and starts flipping switches. It turns on growth genes, things like MYC and Cyclin D1.
So APC is holding back the growth signal by destroying the messenger.
That's the perfect way to put it. It destroys the messenger before the message can be delivered. So the memory hook here is pretty straightforward. APC stands for anti-proliferation controller.
Anti-proliferation controller, or going back to your other analogy, it's the gatekeeper.
It is the gatekeeper.
Yeah.
If the gatekeeper leaves his post, the enemy walks right in.
Which brings us directly to the enemy, or at least the guy who causes all the trouble when the gatekeeper is gone. Player number 2, beta-catenin.
Right. Beta-catenin is fascinating because it actually has a dual life. It sort of moonlights.
Oh.
On one hand, it's a structural protein. It hangs out at the cell membrane and helps cells stick together. It acts like the mortar between the bricks and the colon wall.
So normally, it's a good guy. It's keeping the tissue intact and organized.
Yes, as long as it stays at the membrane where it belongs. But remember, APC is supposed to keep the free-floating beta-catenin in check. If APC is gone, beta-catenin becomes this unsupervised builder.
The foreman left the job site.
Exactly, and it just floods the nucleus and starts building tissue that shouldn't be there. It acts as a transcription factor, treating the DNA like a blueprint for a skyscraper when we only needed a little cottage.
So the dynamic here is absolutely crucial. APC is the brake. Beta-catenin is the accelerator.
Precisely. And the memory hook for beta-catenin is simple. Beta-catenin is the builder protein. If it's not destroyed by the garbage disposal, it just keeps building.
Okay, that makes sense. Next up on our CAS list, we have a famous one, KRAS.
KRAS is huge. It's a proto-oncogene. And what that means is it encodes a signal transduction protein that binds GTP.
Let's just unpack proto-oncogene for a second. That implies it has the potential to cause cancer, but normally it's just a regular part of the cell's signaling network.
Correct. Normally, KRAS is like a relay runner in a complex race. It waits for a growth signal from outside the cell. Maybe a hormone or a growth factor hits the cell surface receptor.
Okay.
That signal then taps KRAS on the shoulder. KRAS grabs a molecule called GTP, which is like the baton, and that turns it on. It then passes the signal down the line to tell the cell to divide.
So it's a switch. It gets flipped on to pass a message.
Yes. But here is the most important part about the normal function. KRAS has a built-in timer. It naturally breaks that GDP down into GDP, which turns it off.
Ah, so it's a self-limiting switch.
It's a self-limiting switch. Click on, signal, click off. It's supposed to be quick.
So it waits for orders, runs its one lap, and then stops to wait for the next order.
Usually, yes. But when it gets mutated, and in colon cancer, this is usually a very specific point mutation at codon 12, it gets stuck in the on state. It loses the ability to turn itself off.
So the relay runner just took off running without the baton and refuses to stop.
Exactly. It's just sprinting around the track over and over again, telling the cell to divide and divide and divide, even though there's absolutely no signal from the outside telling it to do so. The engine is redlining.
The memory hook here has to be in the name itself.
It is. KRAS. Keep running and signaling.
Keep running and signaling. That is perfect. It's the engine that won't idle down. Okay, number 4. This is perhaps the most famous gene in all of cancer biology, TP53.
The legend. The one and only. TP53 encodes the P53 protein. This is a tumor suppressor, but it has a very specific, very high-level job. We call it the guardian of the genome.
The guardian. What is it guarding against?
Damage.
Chaos.
Mutation. P53's job is to monitor the DNA. It hangs out in the nucleus like a smoke detector. If it sees DNA damage, maybe from radiation, maybe just a replication error, it immediately halts the cell cycle. It puts up a roadblock at the G1S checkpoint.
It just yells, "Stop right there. Nobody moves."
Nobody copies their DNA until we fix this mess. And it actually calls in the repair crew, the enzymes that fix DNA. But here is the kicker. If the damage is unfixable, if the repair crew comes back and says it's too far gone, we can't patch this.
P53 makes a hard call.
A very hard call. It induces apoptosis.
Programmed cell death. It basically tells the cell to commit suicide for the greater good.
Exactly. It sacrifices the one damaged cell to save the entire organism. It prevents a cell with broken DNA from replicating and passing on those dangerous errors. But if you lose TP53,
Then you have damaged cells just continuing to divide, passing on their mistakes.
and that is often the tipping point for full-blown malignancy. So the memory hook for P53 is the police officer of the cell. If the police are absent, crime, in this case, mutation, spreads rampant.
Okay, that's a powerful one. We have the gatekeeper, APC, the builder, beta-catenin, the engine stuck on run, KRAS, and now the police officer, P53. Next on our list, we have something called TGF-beta.
Right. TGF-beta stands for transforming growth factor beta. It's a cytokine. Now, the name's a bit confusing because it says growth factor, but in the context of the colon epithelium, it actually inhibits proliferation.
So it's a misnomer. It normally stops growth. It's another brake.
Think of it less like a mechanical brake and more like a traffic light,
Okay.
specifically a red light. TGF-beta is the signal floating around outside the cell. When a receptor on the cell surface sees TGF-beta, it tells the cell machinery to stop dividing.
What if the receptor is broken?
And that receptor is called TGF-br2. If that receptor is mutated or lost, there is no red light. The cell is colorblind to the stop signal. It just runs the intersection.
Got it. Some memory hook. TGF-beta is the traffic light, specifically the red signal.
Exactly.
And finally, the last group of characters on our list, the DNA mismatch repair genes.
The MMR genes. The key players here are MLH1, MSH2, MSH6, and PMS2.
Sounds like a law firm.
Right.
MLH1, MSH2, and Associates.
It kind of is. They are the compliance department. They are the spell checkers. Their entire job is to scan the DNA right after it's been replicated.
Because copying 3 billion base pairs of DNA is a fast process, right? There are bound to be mistakes.
It's incredibly fast. And the enzyme that does the copying DNA, polymerase, it's good, but it's not perfect. It makes typos. And it especially makes typos in areas where the text is repetitive.
Like a stutter.
A stutter, exactly. Imagine trying to type the word banana, but your finger gets stuck on the N key and you type banana. That's what happens in DNA. We call that slippage. The MMR system's job is to spot that extra N and snip it out, to fix a typo before it becomes permanent.
And if they fail, if the law firm is on vacation,
The typos remain. And the next time that cell divides, those typos get copied and new typos appear on top of them. You get this exponential accumulation of errors.
so the memory hook is simply the spell checkers. If they fail, the script gets really, really messy.
You've got it.
Okay, we have our cast, we have the car parts, we have the brakes, the engine, the police, the traffic lights, and the spell checker. Now, let's watch the car crash.
Let's look at pathway number 1.
Segment 2, the APC pathway, also known as the chromosomal instability pathway.
This is the classic pathway.
What is that?
The OG, if you will. When you read about the adenoma carcinoma sequence in textbooks, this is what they are describing. It accounts for about 80% of all sporadic colorectal cancers.
So 4 out of 5 times, this is the culprit.
Yes, and going back to our core question, this is the broken brake model.
The story of the gatekeeper and the builder.
It starts with the loss of the brake. You lose APC, that's the first hit. It's usually a mutation or a deletion on chromosome 5q21.
So the garbage disposal stops working.
Right. And because the disposal is broken, beta-catenin starts to build up. But notice the pacing here. This isn't an explosion. It's a slow accumulation. The car starts to accelerate, but gently. It's just starting to speed up.
And what does that look like in the colon itself?
The tissue starts to thicken. The cells are dividing a little more than they should. You get a small polyp, maybe what we'd call a tubular adenoma.
This is the initiation phase. We have a polyp, but we do not have cancer yet.
Correct. But now that the cell's dividing a bit faster, it is more prone to other accidents. To really get things moving, you need a second hit, and that's usually KRAS.
The engine gets stuck on gas.
Exactly. You get a mutation, usually at 12p12. Now, not only is the brake cut because APC is gone, but the accelerator is floored because KRAS is stuck on. This gives the tumor a massive growth boost.
So the polyp gets bigger.
The polyp gets bigger, it becomes a late adenoma, it starts looking uglier under the microscope, more disorganized.
Okay, so we are speeding up, brake is cut, gas is floored. What's next in the pileup?
Next, you usually lose the traffic light. You lose SMAD, which is part of that TGF-beta pathway we talked about. This is often a deletion on chromosome 18q21.
So now, even if the body tries to send a stop signal via TGF-beta, the driver is just ignoring it.
Completely ignoring it. The car is blowing through all the red lights. And finally, the transition from a benign, albeit ugly adenoma to a malignant, invasive carcinoma usually involves the loss of the police officer.
TP53.
Yes, a mutation at 17p13. The guardian of the genome is gone. Now the genome becomes wildly unstable. The cells are just chaotic. You have cancer.
So the sequence is APC, then KRAS, then SMA, then P53.
AK is P.
And we have a mnemonic for this, right? I think the source material had a good one.
We do. A king sits proudly.
A king sits proudly, APC, KRAS, SMA, P53.
Or, if you want to emphasize the speed of this pathway, because it is a stepwise, slow accumulation over years, even decades, you could say, a kid slowly progresses.
A kid slowly progresses. I like that one even better because it reminds you that this isn't an overnight explosion. This takes years for these hits to accumulate one by one.
That's why screening works.
Right.
You catch the kid before he progresses. You find that polyp at the APC-KRAS stage. You snip it out during a colonoscopy, and you prevent the P53 loss from ever happening. You prevent the cancer.
Now, you mentioned this is called the chromosomal instability pathway. Why that specific term? What's unstable about the chromosomes?
Because the damage here is structural. It's physical. If you were to look at the chromosomes from these cancer cells under a microscope, they would look ragged and broken.
Oh, wow.
They're physically breaking. They're losing huge chunks. You're losing whole genes or even entire arms of chromosomes. It's not just a typo in the book. It's like tearing whole pages out.
And where do we usually see these tumors geographically in the colon? Is there a pattern?
There is. These are typically on the left side of the colon, the distal colon, so the descending colon and the sigmoid colon.
Left side, okay. And are they hot tumors? Is the immune system attacking them?
Generally, no. We call them cold tumors.
Why is that? Why doesn't the immune system care?
Because even though the chromosomes are a mess, the proteins the cells are producing mostly look normal-ish. The cell is just overproducing normal growth proteins. They don't look foreign enough to the immune system to trigger a massive attack, so you just don't see a lot of immune cells inside the tumor itself.
Got it. So pathway 1, to recap, the broken break, AKSP, left-sided, cold tumor, chromosomal chaos.
That's the classic crash.
Now let's switch gears completely. Let's talk about pathway number 2, the MSI pathway.
This is the spell check failure model.
I love this analogy from the notes. Imagine typing your entire life's work without a spell checker.
It's a perfect description. This pathway is completely fundamentally different from the first one. It's not about losing big chunks of chromosomes. It's about the failure of those mismatch repair genes, MLH1, MSH2, and their friends.
So the spell checker software has crashed.
Right. And just like if you were typing incredibly fast without spell check, typos start to appear. But they don't appear just anywhere. They accumulate rapidly in those repetitive areas we talked about, the microsatellites.
Let's drill down on that. Why are microsatellites so vulnerable to typos?
Because they are slippery for the copying machinery. A microsatellite is just a section of DNA where a short sequence is repeated over and over again, like AAAAAA or CACACA.
It's like a stutter in the genetic code.
Exactly. Now imagine you are a scribe from the Middle Ages, copying a book, and you come across a line that just says CA 50 times in a row. It is incredibly easy to lose count. You might write 49 CAs or maybe 51.
Right. Your eyes would glaze over. It's hard to track.
Oh, and that's what happens to the DNA polymerase. It slips. Normally, the mismatch repair team comes in right behind it and says, hey, you missed one CA there, or you added one too many, and it fixes it. But if the repair team is gone,
the error stays.
The error stays. And because these repetitive sequences are literally everywhere in our genome, the errors just explode across the board. This condition is called microsatellite instability, or MSI.
Hence the name of the pathway. And the memory hook here.
MSI can stand for many small insertions, because that's literally what's happening. Tiny insertions or deletions in the DNA, or if you want a more descriptive one, messy script instability. Messy script instability. I like that a lot. But I have to ask the obvious question. How does a typo cause cancer? I mean, losing a break makes sense. Losing a police officer makes sense. But a typo, it seems so small.
It's a great question. And you're right. Typos seem harmless until you realize where they are. It turns out some very important functional genes have these repetitive microsatellite sequences inside their coding regions.
Oh, I see. So if you make a typo in the middle of the instruction manual for a critical gene...
You break the gene. The instructions no longer make sense. Two key examples are TGF-beta and BAX.
TGF-beta. Wait a minute, that's our traffic light receptor again.
Exactly. It has a long, repetitive sequence of adenines in its code. So in MSI tumors, the polymerase slips when copying TGF-beta. The spell checker isn't there to fix it, and suddenly you have a frameshift mutation. The resulting receptor protein is total garbage. The red light doesn't work. Uncontrolled growth. And what about BAX?
BAX is a pro-apoptotic gene. It helps trigger that programmed cell death we talked about with p53.
Yeah.
It also has these repeats. So you get a typo in BAX, and the cell forgets how to die.
Wow. So you have uncontrolled growth, plus a refusal to die. That is a pretty potent recipe for cancer.
It is. This is why we call it the mutator phenotype. The cell just mutates at a hyper speed rate, breaking all of its own safety equipment along the way.
Now, how does this car crash look different from the APC one?
Very, very different. First of all, these tumors tend to be on the right side of the colon, the proximal colon, the ascending, the transverse colon.
Right side. Okay, opposite of APC.
Secondly, they often look mucinous under the microscope. They produce a huge amount of mucus. It's a very distinct appearance.
Gross, but distinctive. Got it.
And here is the most important clinical distinction. These tumors are full of immune cells. We call them tumor-infiltrating lymphocytes, or TILs.
Now, why is the immune system so interested in these tumors? You said the ATC tumors were cold. Why are these ones hot?
Because of the typos. Think about it. In the APC pathway, you're just amplifying normal growth signals. You're making too much of a normal protein. But in the MSI pathway, you're creating thousands of weird, mutated garbage proteins because of all those frame shift errors.
The proteins themselves look weird. They look foreign.
They look like viral proteins or bacterial proteins to the immune system. We call them neoantigens, new antigens.
Yeah.
The immune system's surveillance cells look at these cancer cells and say, wait a minute, you don't belong here at all, and it launches an attack.
That is fascinating. So MSI tumors are hot tumors because the immune system is actively trying to fight them.
Correct. And that has huge implications for treatment down the line. These patients often respond incredibly well to immunotherapy, which basically takes the leash off the immune system and lets it finish the job. But we'll stick to the pathology for now. One last thing on MSI. It's strongly linked to a hereditary condition.
This would be Lynch syndrome.
Yes. Lynch syndrome, or HNPCC, is basically being born with a broken spell checker. Yeah. You inherit a bad copy of one of those MMR genes from your parents. So every single cell in your body is already one step closer to this pathway.
Okay, so pathway 2, the spell check failure, messy script instability, right-sided, mucinous, hot immune response.
You got it. Now for the third pathway, the one that scares the students, the CIMP pathway. The CIMP pathway. This is the gene silencing model.
Why does this one scare people so much? Is it just the intimidating acronym?
The acronym doesn't help, but it's mostly because it introduces the concept of epigenetics. People get comfortable with genetics, mutations, deletions, broken parts, hard physical changes. Epigenetics is softer. It's not about breaking the gene, it's about turning it off.
It's the software, not the hardware.
That is the perfect analogy. CIMP stands for CpG island methylator phenotype.
That is a mouthful. Let's break that down piece by piece. What is a CpG island?
It's surprisingly simple. It is just a region of DNA that is really rich in the bases cytosine and guanine C and G. These islands of CGs are usually found near the promoter of a gene.
And the promoter is like the on-off switch for the gene.
Yes, it's the landing pad for the machinery that comes to read the gene and make a protein. Now, in our cells, we have this ability to add a methyl group, it's just a little chemical tag, a carbon with 3 hydrogens, to these cytosine bases. And when you do that, it acts like a lock.
That is methylation.
That is methylation. It physically blocks the reading machinery from landing on the promoter. It silences the gene. The gene is still there. It's perfectly intact, but it cannot be read.
It's like gluing the pages of the instruction manual together.
Precisely. So in the CMP pathway, the car crash isn't caused by a broken part, but because someone hacked the software and turned off the safety systems. That is a perfect analogy. And specifically, the key gene that gets turned off is often MLH1.
Wait a second. MLH1, that was one of our spell checkers from the MSI pathway.
Bingo. And this is the exact point where students get confused.
Yeah.
CIMP often causes MSI.
Ah, okay, let me see if I have this right. You can get microsatellite instability because the spell checker gene is mutated, the hardware is broken. That's what we see in Lynch's syndrome.
Correct.
Or you can get MSI because the spell checker gene has been methylated, the software is turned off, and that's CIMP.
Exactly. You've nailed it. If the MLH1 promoter gets covered in methyl groups, you stop making the MLH1 protein. You stop making the spell checker. So you end up with microsatellite instability. But the root cause is different. One is a genetic mutation. The other is epigenetic silencing.
That is a crucial distinction. CIMP is the cause. MSI is the result in this particular case.
Correct. And CIMP has a very specific signature that helps us identify it. It is almost always associated with a BRAF mutation.
BRAF, we haven't talked about it much yet.
BRAF is another signaling protein very similar to KRAS. It's in the same signaling chain, just one step down. But here is the key clinical rule. CIMP tumors usually have a BRS mutation, but they very rarely have a KRAS mutation. It's usually one or the other.
They're mutually exclusive. You don't need to have 2 feet on the gas pedal.
Generally, yes. And they also rarely have TP53 mutations. It's a very distinct molecular profile. It's its own beast.
So do we have a memory hook for CIMP?
We do, and it involves that methylation aspect.
No.
CIMP can stand for covered in methylation permanently.
Covered in methylation permanently. Or maybe cancer initiated by methylation process?
Both work. And if you are ever in an exam or a high-stakes meeting and you need the one-line summary, the Viva answer, as they call it,
Lay it on us.
CMEF is BRAF-driven and methylation hidden.
BRAF-driven and methylation hidden. I like that. It captures the driver, BRAF, and the sneaky mechanism, methylation.
So to recap the CMP crash, someone turns off the engine control software. That's the methylation of MLH1. This kills the spell checker. Typos accumulate, MSI. And usually there is a distinct driver, BRAF, pushing the car forward.
Okay. We have analyzed the 3 crashes in detail. Now let's zoom out. Segment 5, the master comparison.
This is where we synthesize it. If you are listening to this and you feel your brain getting full, this is the part to really focus on, visualizing the 3 cars side by side.
Let's line them up. Car crash number 1, the APC pathway.
This is the brake failure. You lose APC, the brake. You step on KRAS, the gas. You drift slowly and erratically into danger until you lose P53 and hit a wall. This is chromosomal instability.
It happens on the left side. It's a cold tumor.
Perfect summary.
Car crash number 2, the MSI pathway.
This is the navigation and spell check failure. Your map is full of typos because the MMR genes are broken. You are making hundreds of small errors everywhere, so the car just starts falling apart.
And the immune system spots your bad driving and tries to pull you over. Those are the TILs. This is microsatellite instability. It happens on the right side. There's a hot tumor.
Couldn't have said it better myself.
And car crash number 3, the CIMP pathway.
The software hack. You don't break the parts. You just silence the safety genes via methylation. This leads to the same spell check failure as car number 2, so you get MSI, but it's driven by BRAF and started by epigenetics, not a primary mutation in the spell checker itself.
That really helps solidify it. It's all about the mechanism of failure.
Exactly.
Now, if a student is in an oral exam and they're asked to summarize this entire field in one breath, the real mic drop answer, what do they say?
They take a deep breath and they say, broadly, 2 genetic mechanisms exist: chromosomal instability via APC mutation and microsatellite instability via mismatch repair deficiency. A subset of the MSI cases arises via CpG island hypermethylation with BRAF mutation.
Boom.
Yeah.
A+ right there. It acknowledges the 2 main roads, which are CIN and MSI, and then it correctly identifies the epigenetic side road, CIMP, as a cause of MSI. It's elegant and it's precise.
Okay. We've done the molecules, we've done the pathways, but this is a disease that affects real people. Let's move to segment 6, clinical reality and epidemiology.
This is the so what section. Why does all this biology matter for the patient walking into the clinic?
First off, just the numbers. This is a big problem.
Huge. It's something like 1.2 million new cases a year globally, but it's not evenly distributed at all.
Well,
No, it is remarkably higher in westernized regions. North America, Europe, Australia. It is much, much lower in Africa and South Central Asia.
that points a huge flashing neon finger at lifestyle and diet.
It absolutely does. The risk factors are the classic Western diet. Low fiber, high refined carbohydrates, high red meat and processed meat, high fat.
So burgers and fries, not lentils and kale.
Essentially. The prevailing theory is that this kind of diet fundamentally alters the gut microbiota, the bacteria in your colon, and it increases the production of secondary bile acids, which can be carcinogenic to the lining of the colon. Now, let's connect the biology to the symptoms. You mentioned right versus left earlier. This is a classic clinical maxim, but I want to understand why it's so consistent.
It comes down to basic anatomy and physics. The colon is not a uniform tube. The right side, the ascending colon, and the left side, the descending and sigmoid colon, are very different environments.
Let's start with the right side. These are the proximal cancers. This is where we see the MSI and CIMP tumors more often.
Right. The right colon is much wider. The diameter is large. And crucially, what is the consistency of the stool inside the colon at that point?
It's liquid.
Yes.
Yeah.
It's mostly a liquid slurry coming from the small intestine. Most of the water hasn't been absorbed yet.
So if a tumor grows there,
It very rarely blocks the pipe. The liquid stool can just flow right past it. Instead, these tumors tend to be polypoid. They grow outward into the lumen like a mushroom or a cauliflower.
okay.
And they are friable, they're fragile, they bleed easily, but they don't block.
So the patient doesn't come in complaining of constipation or a blockage.
No. They come in tired, they look pale, they're short of breath when they walk up the stairs, and their blood work shows iron deficiency anemia because they have been slowly, silently bleeding from this mushroom-like tumor for months.
And this leads to that golden rule of medicine that gets drilled into every medical student.
Yes. Iron deficiency anemia in an older man or a postmenopausal woman is a GI malignancy until proven otherwise.
You cannot just give them iron supplements and send them home. You have to scope them.
You have to look in the right colon, absolutely.
Now let's contrast that with the left side, the distal colon. This is where the APC, the chromosomal instability tumors, usually live.
The left colon is much narrower. The pipe is smaller, and by the time stool gets there, most of the water has been absorbed. It is solid.
So it's a smaller pipe with solid contents moving through it.
Exactly. And the tumors here tend to grow in an annular pattern. They don't grow like mushrooms. They grow like rings. We call them napkin ring lesions because they grow around the entire circumference of the colon and squeeze it shut.
Like tightening a belt around the inside of the intestine.
Yes. So these patients do get obstruction. They get cramping. They get altered bowel habits, constipation, alternating with diarrhea. And a very classic sign, pencil-thin stools.
Because the stool is being squeezed through that tiny constricted opening.
Exactly. So the biology, the MSI versus the APC pathway aligns with the anatomy, the right versus the left, which then dictates the symptoms, anemia versus obstruction. It all connects beautifully.
That is such a helpful distinction. It makes the symptoms logical rather than just a list to memorize. Finally, let's just touch on staging. Because once you find it, you have to stage it to know what to do next.
TNM. T is tumor, N is node, M is metastasis.
What matters most for prognosis?
Depth. The T stage is all about how deep the tumor has invaded into the wall of the colon. Is it just in the mucosa? Has it gone into the muscle? Has it gone all the way through the muscle? If it's just on the surface, you can often just snip it out. If it's through the wall, it's much more dangerous.
And the nodes, the N.
The N stage. Has it spread to the local lymph nodes? If yes, the survival rate drops significantly because it means the cancer has found a highway system to travel on.
And where does it go if it leaves the colon? What's the most common destination?
The liver. That is the most common site of metastasis by far.
And why the liver specifically?
Again, it's just plumbing. It's anatomy. The blood from the entire colon drains into the portal vein, and the portal vein goes straight to the liver. The liver acts like the first filter for everything coming from the gut.
So the liver catches the traveling cancer cells.
It does. After that, I can go to the lung and the bone, but the liver is the big one.
What are the survival rates looking like these days?
In the USA, the overall 5-year survival is about 65%. But that is a huge average. If you catch it at stage 1, when it's still just in the colon wall, it's over 90%. If you catch it at stage 4, when it's already in the liver, it's much, much lower.
Which brings us right back to the incredible importance of screening. But that's a whole other deep dive for another day. All right, we have covered a massive amount of ground. We've unlocked the code. We've looked at the machinery. Let's try to wrap this up.
Let's do a final recap.
If the listener walks away with only 3 concepts today, just 3 things to hold on to, what should they be?
1. APC.
Yep.
The gatekeeper is lost. The brake fails. The car crashes slowly due to chromosomal instability. That's the classic pathway.
2. MSI. The spell checker is lost. The repair team is gone. Typos accumulate in the map, causing microsatellite instability. That's the Lynch pathway.
And 3. CIMP. The genes are silenced. The software is hacked by methylation, which then often leads to MSI. That's the epigenetic pathway.
So create spell check, software.
That's it. If you have those 3, you have the framework for everything.
And I want to leave everyone with a provocative thought to chew on. We talked about the Western diet and lifestyle, and we talked about CIMP being caused by methylation, these little chemical tags being added to DNA. It just makes you wonder. Those methyl groups have to come from somewhere. Our diet, our metabolism, our microbiome, they provide the raw materials for these chemical tags. Things like folate from leafy greens are critical.
That is absolutely the frontier of this research. How much of this molecular machinery is being directly triggered by what we ate for lunch? We know diet affects the microbiome. We know the microbiome produces metabolites that affect inflammation and cell turnover. It directly connects the microscopic, a single methyl group on a gene promoter, to the macroscopic burger on a plate.
It's a sobering but also an incredibly empowering thought. We aren't just passive victims of our genes, we are in a constant dialogue with them.
We absolutely are.
Well, on that note, I hope next time you hear colorectal cancer, you don't just see a scary mass, you see a car with a broken brake or a scribe with a messy script.
A kid slowly progresses.
Versus messy script instability. I'm definitely using those. Thanks for breaking this all down with us.
My pleasure. It was fun.
And thank you for listening to the deep dive. Keep those neurons firing, and we'll see you next time.